Regulation of respiration. Breathing is normally an involuntary process, but sometimes it can be voluntary also. Breathing movements are controlled, ventilation rate is normalized, and the basic respiratory rhythm is established through the negative feedback impulses from the respiratory receptors to respiratory centers. This regulation involves rhythmic generation of impulses within the brainstem and their transmission to respiratory muscles. This inherent rhythmicity arises within the medulla oblongata. It may be influenced by the impulses from pons varolii, hypothalamus, cerebral cortex, and the 9th and 10th cranial nerves. And respiratory receptors. In human beings, there are two sets of respiratory receptors. In human beings, there are two sets of respiratory receptors, namely structure receptors or Herring-Brewer receptors and chemoreceptors. Structure receptors are located on the walls of trachea, bronchi, and bronchioles. Chemoreceptors include the medullary receptors, aortic bodies, and carotid bodies. These are located respectively in the medulla oblongata, wall of aorta, and the wall of carotid sinus, dilated region of internal carotid artery. So, chemoreceptors are sensitive to changes in the O2 and CO2 levels in blood, especially to low O2 and high CO2 levels. On the other hand, structure receptors are sensitive to high O2 and low CO2 levels, and also it stretches in the lungs. The feedback impulses from structure receptors are concerted with the mechanics of pulmonary ventilation, and those from the chemoreceptors are concerned with the rate and depth of pulmonary ventilation. So, respiratory centers and respiratory rhythm. The respiratory centers control the mechanics of breathing. During breathing, respiratory muscles contract and relax in response to the impulses from the respiratory centers in medulla oblongata and pons varolii. In human beings, there are two sets of respiratory centers, namely medullary centers or medullary rhythmicity areas and pontine centers. Medullary centers are located in medulla oblongata and pontine centers in pons varolii. There are two medullary centers, namely the inspiratory center or dorsal respiratory center and expiratory center or ventral respiratory center. So, pontine centers include the pneumotaxic center and apneustic center. Pneumotaxic center is a inhibitory in function and all other areas are excitatory. Inspiration occurs when the O2 levels in the blood falls low and the CO2 level goes high. Chemoreceptors perceive this stimuli and transform them as sensory impulses. These impulses are transmitted to the inspiratory center via sensory neurons. On receiving them, the inspiratory center gets stimulated and generates motor impulses. These impulses are transmitted to the inspiratory muscles, external intercostal muscles, and the muscles of diaphragm. They stimulate the inspiratory muscles and thereby set the basic respiratory rhythm. Now, inspiratory muscles contract, leading to the expansion of the thoracic cavity and the dilation of the lungs. This brings about inspiration. At the end of inspiration, the inspiratory muscles become inactive and the whole cycle of events goes on repeating over and over again. So, expiration occurs after the lungs have dilated, the O2 levels of the blood has gone high, and the CO2 level has gone low. Structure receptors perceive the stimuli and transform them as sensory impulses. These impulses are transmitted to the expiratory center along the vagus nerve, 10th cranial nerve. The expiratory center responds to them by transmitting inhibitory impulses to the inspiratory center. This inhibits the inspiratory center, leading to the relaxation of the inspiratory muscles, the compression of thoracic cavity and lungs, and the consequent expiration. So later, expiratory center in fact regulates inspiration as well as expiration during normal or quiet breathing. It remains inactive without any role in regulating the respiratory rhythm. This is because in quiet breathing, inspiration is accomplished by the active contraction of inspiratory muscles, and expiration results from their relaxation and the passive recoil of lungs and thoracic wall. But in intense or forceful breathing, some of its numerous nerves control inspiration and others control expiration. So, in quiet breathing, inspiratory and expiratory centers are never active simultaneously, but in forceful breathing, they act almost together. At the beginning of expiration, the inspiratory center remains inactive, but after expiration, it suddenly and automatically becomes active. Then, pneumotaxic centers exerts periodic checks on the inspiratory and apneustic centers. Its signals are transmitted to the switch off points of the inspiratory area. The signals temporarily turn off the expiratory center, limit or suspend inspiration, and thereby facilitate expiration. When the signals are strong, inspiration becomes short living, lasts only up to 0.5 seconds, and the lungs are only partially filled. When the signals are weak, inspiration becomes long lasting, lasts up to 5 seconds, and the lungs get completely filled. In effect, strong pneumotaxic signals shorten inspiration and expiration and thereby increase the breathing rate. On the other hand, weak signals prolong inspiration and expiration and thereby lower the breathing rate. So, apneustic center sends impulses to the inspiratory center. This activates the inspiratory center and thereby prolong inspiration and inhibits expiration. So, apneustic center and pneumotaxic center appear to be antagonistic in function. In quiet breathing, inspiration takes nearly 2 seconds and expiration takes 3 seconds. This is the basic rhythm. At the beginning of inspiration, the inspiratory center becomes active for 2 seconds. This stimulates the inspiratory muscles and causes inspiration. After 2 seconds after inspiration, it becomes inactive for 3 seconds. This causes the relaxation of inspiratory muscles and results in expiration. Very soon it becomes active again for the next inspiration. This turning on and off of the inspiratory center is controlled by the pneumotaxic center. Then, cortical influence. Respiratory centers have some connection with the cortical cortex, cerebral cortex. This enables a person to voluntarily alter the pattern of breathing and even to stop breathing for a short while because connections with cerebral cortex. Voluntary control is protective in that it enables to prevent the entry of water, irritating gases, etc. into lungs. However, the ability to stop breathing is limited by the buildup of CO2 in the blood. When the PCO2 goes beyond a certain level, the inspiratory center will be stimulated. Impulses will be sent to the inspiratory muscles and breathing will resume whether or not the person wishes. So it is impossible for a person to commit suicide by voluntarily holding his breath. Chemical stimuli and control of ventilation rate. There are certain chemical stimuli which determine how fast a person can breathe. They help to maintain the proper levels of oxygen and carbon dioxide in the blood and also to respond to the changes in the blood levels of these gases. An increase in CO2 level will cause an increase in H plus ions and a decrease in CO2 level will cause a decrease in H plus ions. The main chemical stimulus that regulates breathing rate or ventilation rate is the high CO2 level in the blood. High CO2 level or hypercapnia or hypercarbia stimulate the aortic and carotid chemoreceptors. They generate sensory impulses and transmit them to the inspiratory center. The inspiratory center gets highly excited, generates excitatory motor impulses and transmits them in rapid release to the inspiratory muscles, extended intercostal muscles in between ribs, and phrenic muscles in the diaphragm. This muscle contract and relax rapidly, increasing the breathing rate. The increased breathing rate is called hyperventilation. This allows the body to expel more CO2 until the blood levels of CO2 or PCO2 falls to normal. Hyperventilation stimulates the stretch receptors on the trachea, bronchial and bronchiolar walls. They generate sensory impulses and transmit them to the expiratory center. Expiratory center gets excited and transmit inhibitory motor impulses to inhibitory muscles. inspiratory muscles. This temporarily inhibits inspiratory activity and thereby prevents sustained inspiration or apneusis. When inspiratory muscles relax, leading to expiration, soon the expiratory center becomes inactive and the inspiratory center becomes active again. This respiratory cycle goes on repeating rhythmically throughout life. When the blood level of CO2 is lower, apokenia or apokabia, the chemoreceptors will not stimulate it and no impulse would be transmitted from them to the inspiratory center. Consequently, the inspiratory center will set its own moderate pace and rhythm until CO2 accumulates and the PCO2 rises to the normal. This causes slow breathing rate called hyperventilation. Then the chemoreceptors for oxygen are stimulated only to subnormal levels of PO2. Under such conditions, they get stimulated and transmit impulses to the inspiratory area. If the fall in PO2 are abnormally low, the neurons of the inspiratory center will suffer from oxygen starvation and will not properly respond. They will send out fever impulses to the inspiratory muscles. Consequently, the breathing rate falls abnormally low or breathing stops, apnea. Then inspiratory problems in newborn babies. The respiratory system of normal newborn babies is fully developed and perfectly functional, but in some abnormal cases, newborn babies suffer from severe respiratory problems. Infant respiratory disease syndrome or IRDS, sudden infant death syndrome or SIDS, and the aphasia neonatorum are such major problems. First, infant respiratory disease syndrome. Distress syndrome is respiratory distress syndrome or RDS is a respiratory disease of adults and newborn babies. Adult RDS is a complication of shock, systemic sepsis, viral respiratory infections, etc. It is characterized by pulmonary edema with damages to pulmonary mucosa. Despite the advancement in medical science, it still remains as a serious disease and nearly 50% mortality cause in babies. The lungs of RDS patients are surfactant deficient. Surfactant is a mixture of lipids, proteins, and glycoproteins. It is produced in specialized lung cells called type 2 pneumocytes. In the cells, it gets packaged into lamellar bodies, which are soon extruded to the alveoli. In the alveoli, the lamellar bodies unfold and form a complex lining of the alveoli. This lining prevents the sticking together and collapse of alveoli walls towards the end of expiration. Infant RDS, primarily called hyaline membrane disease or HMD or glassy lung disease GLD, is a fatal respiratory problem of newborn babies. It is found in premature infants, in the infants born from cesarean section in which infants of diabetic mothers, especially when diabetes is untreated or poorly controlled, and also in the second born of premature twins. The main feature of infant RDS is developmental insufficiency of surfactant production and the structural immaturity of lungs. Surfactant is a surface active agent lining the alveoli and lungs. It plays an active role in active normal respiration. It allows the alveoli to retain open throughout the inspiration expiration cycle for gas exchange. Also, it prevents the alveoli from sticking together and collapsing at the end of expiration. Its deficiency or absence is one of the factors responsible for infant RDS. Infant RDS begins in a few hours after birth. It is manifested by difficult and labored breathing, trachypnoea, and chest pain contraction during breathing efforts, ranting expiration, flaring nostrils, cyanosis, blue coloration, etc. are also common symptoms. Infant respiratory distress syndrome. As the disease progresses, the baby may suffer from ventilation failure, hypercapnia, and prolonged apnea. So complications of the disease include acidosis, low blood sugar level, low blood pressure, chronic changes in lungs, intracranial hemorrhage. The renal course of the disease may last for 2 to 3 days. In most cases, death may occur soon after the onset of the disease or may be delayed for a few days. In some instances, the child may survive. Then RDS affects nearly 1% of the newborn babies with more than 50% of mortality rate. It is one of the leading causes for the death of preterm infants. It is perhaps the commonest single cause for the death of the first month of postnatal life. Despite the spectacular advancements in medical science, a promising result in the treatment of RDS is still elusive. Administration of glucocorticoids to the mother at least 12 hours before delivery may sometimes prevent it or reduce its incidence. It probably speed up the production of its surfactant in the infant. A treatment called positive and end respiratory pressure is now given to RDS patients. It involves the principal lines enable supply of oxygen-rich air through the endotracheal breathing tube with constant positive airway pressure or to off-beat apnea, administration of synthetic or extracted surfactant to the lungs through the breathing tube to make sure surfactant deficiency. Then intravenous administration of fluids to stabilize blood sugar level, blood electrolyte level, and blood pressure level. One of the most commonly used exogenous surfactant is Cervanta, extracted from cow lungs. It can reduce the death rate by 30%. Next is SIDS or sudden infant death syndrome. SIDS, also called crib death or cot death, is the unexpected, sudden and silent death of an apparently healthy baby, usually during sleep. It kills thousands of babies every year. The unfortunate victims are aged between 1 week and 12 months. The commonest feature of the disease is the exudation of the blood-tinged froth from the nostrils. For centuries, cot death was mistaken due to suffocation. Although several causes have been attributed to this fatal disease, the actual cause is not still really known. However, death is most causes result from laryngospasm, believed to be triggered by a viral infection of the upper respiratory tract or from prolonged apnea due to the malfunctioning of the respiratory center. Then Asphyxia neonatorum. Asphyxia neonatorum is the infant asphyxia. Infant asphyxia, characterized by imperfect breathing or difficult breathing in the newborn infant. It contributes to nearly 50% of infant death during the first month of life. It is mainly due to an interference with the oxygenation of blood. Its common causes include serious illness of the mother during the later months of pregnancy, anesthetics or analgesics given to the mother during labor, prolonged labor, obstruction to the blood circulation in the umbilical cord, obstruction in the air passage of the infant, intracranial hemorrhage, congenital heart disease, etc. The onset of infant asphyxia occurs before or after birth, so it can prenatal and intrauterine or postnatal and extrauterine. So prenatal asphyxia could be due to failure in the proper supply of oxygen to the fetus before or after labor, whereas postnatal asphyxia may be mostly due to the prematurity or congenital problems of the child. Some of the commonest causes of the prenatal asphyxia as are the following: prolonged and repeated uterine contraction during labor, which may interfere with gas exchange in placenta. Unequal pressure excreted by the uterus. Exerted by the uterus on placenta and umbilical cord. This may prevent the free passage of oxygen to the fetus. Compression or tearing of placenta, twisting of or tearing of the umbilical cord, premature partial or complete separation of the placenta, then maternal anemia or asphyxia. In extrauterine asphyxia, the baby attempts for breathing but does not succeed satisfactorily. The reason for this failure include the presence of blood or mucus in the respiratory tract, anomalies of heart or lungs, skull injury, intracranial hemorrhage, inherent weakness of respiratory muscles, improper functioning of the respiratory centers, etc. Asphyxia neonatorum is of two types, namely asphyxia levita and asphyxia pallida. The former is asphyxia of the first grade and the later is asphyxia of the second grade. In asphyxia levita, the skin turns blue or reddish blue, face becomes swollen, eyes protrude, then heart beats strongly and breathing effort may be absent or rather weak and infrequent. In asphyxia pallida, vasomotor center goes overstimulated due to high levels of blood CO2. Practically, there is no attempt for respiration or there may be feeble efforts in some cases. Heartbeat may be weak. The unfortunate baby will have a corpse-like appearance. Only the visible signs of heartbeat and a few breathing gasps show that the baby is not dead. The treatment of infant asphyxia involves the cleaning of the respiratory passage and supply of oxygen to tissues. In mild cases, the baby is held in an inverted position and a finger is gently introduced into its throat. It may remove the obstruction. In asphyxia livida, there is usually mucus in the trachea or bronchi. It must be frequently removed by inverting the child and introducing a catheter into the glottis. Once the air passage is cleared, external irritation must be tried to reflect stimulation of respiration, slapping on the back and buttocks of the inverted baby, sprinkling of cold water over the body, rubbing over the body with warm cloth. Some common measures applied in mild cases. In severe cases, the child is immersed in warm water for a few minutes and then in cold water. Warm bath relieves the vasoconstrictor spasm and other overloaded heart. Weak mustard bath and careful compression of the chest are also tried. In extremely severe cases, artificial respiration is essential. Then respiratory problems in old age. Aging adversely affects respiratory efficiency and causes several respiratory problems. Aging lungs and airways become less elastic and less effective, severely affecting pulmonary ventilation. As a result, a good amount of inspired air remains trapped in the airways, considerably lowering the intake of usable air into the lungs. In other words, there would be a progressive increase in the volume of dead air and the progressive decrease in ventilation rate as aging progresses. Dead air is a portion of the air, inspired air retained in the airway. The commonest age-related respiratory problem include pneumonia, obstructive lung conditions, tuberculosis, etc. First is about pneumonia. Pneumonia is an acute infection and inflammation of the alveoli of the lungs. Pulmonary inflammation caused by allergic reactions is called alveolitis, and that caused by chemical or physical agents is called pneumonitis. The organisms causing pneumonia include virus, bacteria, mycoplasma, fungi, and protozoa. In healthy persons, it may be caused by Streptococcus pneumoniae, Mycoplasma pneumoniae, or a virus. The causative organism of pneumonia following influenza is Streptococcus aureus. The major symptoms of pneumonia are fluid-filled alveoli containing dead leukocytes, low oxygen level in blood, hypoxia due to low rate of gas exchange in lungs, harsh and high-pitched breathing, cough, and fever. In pneumonia, the alveolar sacs fill up with fluid and white blood cells, considerably reducing the air sac inside the lungs. Oxygen has great difficulty to diffuse through the inflamed alveoli, and so the O2 levels in the blood may fall abnormally low. At the same time, the blood CO2 level remains normal since CO2 can diffuse through the inflamed alveoli more easily than O2 can. The common symptoms of pneumonia include cough, fever, pleuritic pain, dysphoia, cyanosis, etc. Formerly, pneumonia was classified into three types, namely lobar pneumonia, lobular or segmental pneumonia, and bronchopneumonia. So, in lobar pneumonia, all the alveoli of a lung lobe are inflamed. In lobular pneumonia, the alveoli of only a portion of a lobule of a lobe are inflamed. And in bronchopneumonia, alveoli and bronchiolar tubes are inflamed. This difference has no relevance now. Obstructive lung conditions. These are the conditions which construct a limit airflow through the airway in aged persons. Some of such conditions are as for the following: build-up of naturally occurring mucus. This is quite common in chronic obstructive pulmonary disease (COPD). This mucus accumulation is only partially reversible, and so it considerably blocks the flow of air, leading to respiratory problems. Then disruption of the support tissue around airway. Old age wear and tear naturally disrupts the passage package tissue around airways. This eventually leads to the local collapse of airways, resulting in emphysema. Then tightening of the lung muscle around airways. This constricts airways, leading to severe breathing difficulties, asthma, etc. Then thickening and hardening of the walls of airways. Thickening and hardening of the walls of airways by fibrosis reduces the lumen of the airway. It also adversely affects the free flow of air. Then respiratory problems and adaptation at high altitudes. Respiration at high altitudes is always associated with some problems. This is mainly because of three major reasons, namely very low barometric atmospheric pressure, insufficiency of O2, and low partial pressure of atmospheric O2. Atmospheric pressure and the percentage of partial pressure of atmospheric O2 decrease with increased altitude. Decrease in atmospheric pressure often causes dysbarism or decompression thickness. In this case, nitrogen and some other dissolved gases bubble out from blood due to a sudden fall in their pressure. It may result in severe body pain, muscular paralysis, unconsciousness, and even death. Decomposition sickness is common among deep divers and miners also when they suddenly come to the surface. In human beings, decrease in the partial pressure of O2 at high altitudes often causes arterial hypoxia. Hemoglobin can easily combine with O2 only at high PO2. So the low PO2 at higher altitudes adversely affect the uptake of O2 by hemoglobin, resulting in hypoxia or anoxia. Periodic breathing and mountain sickness. Prolonged and severe hypoxia at high altitudes may cause periodic breathing and mountain sickness. Periodic breathing is characterized by a recurring pause between successive breaths. Every pause may last 5 to 10 to 20 seconds. This considerably reduces the breathing rate and the intake of O2. So, respiration at high altitude is always associated with some problems. This is mainly because of three major reasons, namely very low barometric or atmospheric pressure, insufficiency of O2, and low partial pressure of atmospheric O2. Atmospheric pressure and the percentage and partial pressure of atmospheric O2 decreases with increasing altitude. Decrease in atmospheric pressure often causes dysbarism or decompression sickness. In this case, nitrogen and some other dissolved gases bubble out from blood due to a sudden fall in their pressure. It may result in severe body pain, muscular peristalsis, paralysis, unconsciousness, and even death. Decompression sickness is common among deep divers and miners also when they suddenly came to the surface. In human beings, decrease in the partial pressure of O2 at high altitudes often cause arterial hypoxia. So hemoglobin can easily combine with O2 only at high PO2. So the low PO2 at higher altitudes adversely affects the uptake of O2 by hemoglobin, resulting in hypoxia or anoxia. Periodic breathing and mountain sickness. Prolonged and extreme hypoxia of higher altitudes may cause periodic breathing and mountain sickness. Periodic breathing is characterized by recurring pauses between successive breaths. Each pause may last for 5 to 10 or 20 seconds. This considerably reduces the breathing rate and the intake of O2. Mountain sickness or altitude sickness is a group of symptoms which appear when people reach great heights either on mountaineering or in air travel. Considerable shortage of oxygen in the inspired air is the main cause of it. So, mountain sickness is of two types, acute and chronic. The former frequently affects people of several days after arriving at altitudes above 3,000 meter. It is characterized by mental fatigue, physical exhaustion, hallucinations, drowsiness, giddiness, unconsciousness, convulsions, headache, nausea, vomiting, dyspnea, bluish skin, lips and nails, etc. It may sometimes lead to coma and death. Chronic mountain sickness occasionally results from long-term stay at night, high altitudes. It is also characterized by fatigue, exercise intolerance, etc. Then adaptations at higher altitudes. High altitude animals adjust themselves to hypoxic conditions by responsive mechanisms which raise the red cell count, hemoglobin content, and the oxygen carrying capacity of blood and maintain the high oxygen affinity of hemoglobin. In high altitude animals such as llamas and vicuñas, hemoglobin has much greater affinity for oxygen than in their low altitude relatives. The adaptive physiological response at high altitude fall under two major categories, namely accommodation and acclimatization. Accommodation involves the initial adjustments of the body in response to hypoxia. It includes hyperventilation of lungs, tachycardia, and increased concentration of diphosphoglycerate (DPG) in RBCs. Acclimatization involves changes in body in response to long-term exposure to hypoxia, polycythemia, high red cell count, increases enzyme activity, etc., and some other changes. Then hyperventilation. So altitudinal hypoxia stimulate peripheral chemoreceptors, which in turn stimulate the inspiratory center and thereby increase the rate and depth of breathing. Increased ventilation expels large quantities of CO2. This reduces the CO2 buildup in the body and causes alkalosis. Alkalosis limits the increase in breathing. Alkalosis is gradually corrected by the renal excretion of the bicarbonate. Then tachycardia. Hypoxia increases the heart rate and cardiac output as a responsive reflex. This may enhance the delivery of oxygen to tissues. In human beings, cardiac output may return to normal after about 2 weeks of stay at higher altitude. Then increased DPG concentration. Hypoxia and alkalosis causes an increase in the concentration of DPG within RBCs. This in turn raises the tissue PO2 to the normal level. Then high red cell count. Hypoxia and decrease in blood PO2 promote the excessive production of erythrocytes. Polycythemia. In this case, oxygen deficiency renal cells release an enzyme called renal erythropoietic factor. It acts upon oxygen globulin and converts into a hormone called erythropoietin. This hormone circulates through the blood to red marrow and stimulates erythropoiesis, formation of erythrocytes. This eventually raises the red blood count and hemoglobin content of the blood to counter hypoxia. Erythropoietin is produced in other tissues also, particularly in liver. Then increased enzyme activity. Hypoxia often results in increased enzyme activity and higher cellular diversity of formation of hypoxia. This eventually raises the red blood cell count and hemoglobin content of the blood to counter hypoxia. Erythropoietin is produced in other tissues also, particularly in the liver. Then increased enzyme activity. Hypoxia often results in increased enzyme activity and higher cellular density of mitochondria. This changes increase the ability of cells to function at low PO2. Then summary of physiological adjustments at higher altitudes. The major physiological response to adjust the body to altitudinal hypoxia can be summarized as follows. Altitudinal hypoxia increase the CO2 and decrease the PO2 in the blood. High PaCO2 stimulates the aortic and carotid chemoreceptors, which in turn stimulate the inspiratory center. As a result, ventilation rate increase considerably. Hyperventilation leads to the excessive loss of CO2 from blood. As a result, blood pH increases and pCO2 decreases with a shift in the alkaline direction. This increase in the alkalinity of blood is called alkalosis or alkalemia. High pH and low pCO2 levels in the blood inhibit the chemoreceptors, leading to a fall in the ventilation rate and oxygen supply. Again, the PO2 it decreases and PCO2 increases in the blood, and the whole cycle of events goes on repeating. Simultaneously, erythropoietin stimulates and narrow to produce and release more and more erythrocytes to resist hypoxia. When all these adaptive mechanisms or adjustments are completed, the body becomes acclimatized to higher altitudes. Since this acclimatization process is gradual, mountaineers do not climb mountains at a stretch. Some people and some animals have better acclimatization to higher altitudes than others. The natives of and Himalayas represent select genetic variations in this respect. Then, problems and adaptations of diving mammals. And after that, we need to learn something about respiratory problems and health hazards caused by smoking and artificial ventilation, heart-lung machine. These are the major portions we will going to cover in this session. So, respiratory problems and adaptations of diving mammals. Many lung-breathing aquatic vertebrates are efficient divers, and they can survive long periods of submergence without coming up for air. This is by virtue of their axillary respiratory mechanisms, minimal oxidation requirement, and specialized metabolic patterns. Such vertebrates include amphibians, turtles, crocodiles, aquatic snakes, penguins, cormorants, and aquatic mammals such as whales, walruses, porpoises, seals, beavers, etc. So this is by virtue of their auxiliary respiratory mechanism, minimal oxidation requirements, and specialized metabolic patterns. Such vertebrates include amphibians, turtles, crocodiles, aquatic snakes, penguins, cormorants, and aquatic mammals such as whales, walruses, porpoises, seals, beavers, etc. These animals are adapted to dive for food, so to escape from their enemies. Their ability to remain underwater depends on their ability for maximum storage and minimal utilization of oxygen. Usually these animals depend on the oxygen they obtain periodically from the surface air and use most sparingly underwater. While underwater, they use the atmospheric oxygen already stored in myoglobin. To replenish the used oxygen, they frequently come to the surface. Their capacity for the most judicious use of their oxygen store is astonishing. For example, sperm whales can go as deep as 1,000 meters or more and can remain there for more or more hours without any anoxic effect. The major problems generally experienced by diving mammals are lack of oxygen availability at depths, difficulty for the elimination of CO2 from tissues, and difficulty for the proper distribution of O2 in other different parts, the depletion of the oxygen storage, and the resulting hypoxia and anoxia, bradycardia, and low heart rate. Then slow blood circulation to organs other than brain, heart, and adrenal glands, oxygen toxicity, and gas bubbling due to decompression sickness when the animals ascend to the surface quickly. To overcome these problems, they have special adaptations also. The volume of their blood is generally 2 times greater than that of non-divers. Also, many of them have high concentration of myoglobin and high oxygen storing capacity. However, their lungs are not larger than those of non-divers. During diving, circulatory and respiratory adjustments occur to ensure the proper distribution and maximum utilization of oxygen. The danger of hypoxia or anoxia encountered by the underwater animals is essentially the same as that encountered by the animals at high altitudes. However, the solution is different in two habitats. In high altitudes, the problem is solved by enhancing the rates of oxygen delivery to tissues, while in water, the solution involves the temporary storage and judicious use of oxygen and the capacity to deal with oxygen depth. At the beginning of diving, a set of physiological mechanisms get activated. They collectively serve as the diving reflex. Now breathing stops and the animal makes use of the oxygen stored in the myoglobin. This causes a reduction in the supply of oxygen to most tissues. As a result, heart rate beat becomes weak and slow. Heart rate falls to nearly 1/10 of the normal rate. Total blood circulation gets considerably reduced and blood pressure start to drop. Fall in blood pressure will be prevented by the constriction of some blood vessels. The partial constriction of some vessels and the total constriction of some others cause the redistribution of blood in such a way that its major share goes to vital organs such as heart and brain, and at least withstand which can least withstand hypoxia. Circulation to skin, muscles, and viscera may get sharply reduced. This adaptive adjustment minimizes the utilization of oxygen. At the same time, it ensures a steady supply of oxygen to organs which are highly sensitive to hypoxia. After some time, the oxygen stores in the myoglobin gets exhausted and muscles resort to anaerobic metabolism. This leads to the accumulation of lactic acid in muscles and the building up of oxygen depth. After some time, the animal returns to the surface and breathes in air. As a result, oxygen supply to tissues goes high, heart rate becomes normal, blood circulation to all parts increase, oxygen stored in myoglobin steadily builds up, muscles return to aerobic metabolism, lactic acid gets aerobically oxidized, and oxygen depth is repaid. Then oxygen depth. Oxygen depth is a physiological state in which there is an increased demand for oxygen. It occurs when a normally aerobic animal is forced to respire anaerobically. Consider situations of hypoxia. To meet the body's increased demand of energy, pyruvate is anaerobically converted to lactic acid. Lactic acid is toxic and its breakdown requires oxygen. So accumulation of lactic acid builds up an oxygen depth. The oxygen depth is repaid when oxygen is available again. The lactic acid is oxidized in liver. Then decompression sickness. Decompression sickness or casein disease or dysbadism is a serious condition of gas bubbling from blood. It occurs when a deep water diver or a casein worker, a person who makes tunnels under rivers, suddenly turns to the surface from deep water. In the deep water, pressure is very high, and so large quantities of nitrogen and other gases dissolve in the blood of the diver. Excessive amounts of dissolved nitrogen and body fluids may cause giddiness and other symptoms similar to alcoholic intoxication. This condition is called nitrogen narcosis. The greater the depth, the more severe would be the condition. That is called decompression sickness. In deep water, divers ascends to the surface slowly. The dissolved nitrogen may be eliminated from his body through lungs. However, when he comes to the surface, all of a sudden this occurs a rapid fall in the pressure of the ambient gases in the body. As a result, he suffers from decompression sickness. Nitrogen and other gases bubble out from his blood. In human being, it may block pulmonary circulation and may cause difficulty breathing or respiratory failure, joint pain, dizziness, convulsions, seizures, cardiac arrhythmia, irregular rhythm of heartbeat, myocardial damage or anginal pain, then extreme fatigue, paralysis, and unconsciousness. Usually, the symptoms of decompression sickness begins to appear ultimately or after a short latent period of a few hours. The onset of the problem is acute pain around major joints or in the joint. These persons are more sensitive to fall in pressure than thin and lean persons. Obese persons are more sensitive to fall in pressure than thin and lean persons. So decompression sickness can be prevented by slow ascent or by the use of special decompression tank within 5 minutes after reaching the surface. Use of helium oxygen mixers instead of nitrogen containing air is also suggested, since helium is only about 40% as soluble as nitrogen in blood. Then adaptations of diving mammals. Diving mammals are able to remain underwater for long periods by virtue of the following adaptations: storage of oxygen in the myoglobin, and venous blood reservoir, low metabolic rate and low oxygen requirement, low heart rate and low cardiac output, peripheral vasoconstriction and circulatory shunts to maintain blood flow to brain, heart, adrenal gland, etc., large blood volume and high oxygen carrying capacity of blood, low sensitivity of the respiratory and circulatory centers to increase concentrations of CO2 and lactic acid, tolerance to oxygen depth in muscles, and respiratory problems and health hazards caused by smoking. Smoking has now become a popular habit. It has gifted mankind with several serious diseases which usually take heavy tolls of human life. Cancers, pulmonary diseases, heart ailments, and other cardiovascular diseases are a few of these maladies. Then tobacco smoke. Tobacco is the dried leaf of the plant Nicotiana tabacum. This is used for smoking and also as a stuff. Tobacco smoke is a mixture of more than 1,000 kinds of chemical constituents. The commonest and the more noxious ones among them are nicotine, carbon monoxide, hydrogen cyanide, arsenic, phenol acids, alkalides. Indian tobacco additionally contains toluene. Often these constituents condense to form a thick and brown smoke tar. Several constituents of tobacco smoke are tissue irritating, some are carcinogenic, cancer inducing, and still others are co-carcinogenic, like intensify the action of carcinogens. Nicotine is a poisonous alkaloid responsible for narcotic and soothing effects of tobacco. In mild doses, it stimulates the smoker, but in high doses, it abnormally excites brain centers, accelerates heart rate and pulses, boosts blood pressure, and elevates blood sugar level, and clumping of blood particles, platelets, and their adherence to arterial walls is yet another complication. So, carbon monoxide. What is happening? High doses abnormally excites brain centers, accelerates heart rate and pulse, boosts blood pressure, and elevates blood sugar level. The clumping of blood platelets and their adherence to arterial wall is yet another complication. Carbon monoxide is a product of incomplete combustion in blood. It causes carbon monoxide poisoning. Too much of CO in pregnant women may cause miscarriage or fetal abnormalities. So, hydrogen cyanide is yet another poisoning gas of tobacco smoke. It can upset energy metabolism. Trace amounts of it in heart patients may cause serious consequences and harmful effects of smoking. Long ago, tobacco was considered to be a wonder drug, but at present there are accumulating evidence to show that almost all vital organs are seriously harmed by smoking. Smoking considerably reduces our life expectancy by poisoning our blood, damaging our liver, lung, heart, and kidneys, and also by inducing a host of ailments. Some of the harmful effects of smoking are as follows, like damaged lungs, liver, kidneys, and heart, induces cancers of mouth, throat, lung, liver, stomach, intestine, kidney, bone marrow, inflicts peptic and duodenal ulcers, causes chronic asthma, bronchitis, pneumonia, and emphysema. Emphysema is obstruction of alveolar walls with the result that the number of air chambers decrease, the surface area for gas exchange become much reduced, and the efficiency of lungs gets severely hampered. Then induces obstructive pulmonary diseases and thereby reduces the functional efficiency of lungs, lowers the sensitivity of olfactory epithelium and injuries, inactivates or kills the defensive cells of the airway and lungs, paralyzes the ciliary activity in the air tract, and promotes excessive secretion of mucus. Then aggravates cardiovascular diseases and cirrhosis of liver. Then excessive smoking by pregnant women may cause abortion, miscarriage, stillbirth, and infant deformities. And suppressed sexual drive, decreases sperm production, and seriously impair reproductive powers. Smoking may cause gangrene, necrosis, or mortification of a part, such as toe, due to the blockage of the veins or artery which carries blood to that part. It often necessitates the amputation of the affected part. Everybody is now fully aware of the tragic fate that awaits a smoker. Even then, people continue to blissfully smoke away their life. Now we are going to learn some methods to support, artificial ventilation, respiration helpers, such kind of stuff. So aspirator is one of them. Aspiration is the withdrawal or sucking out of fluid or gases from the cavities of body or from the cavities produced by diseases. That is done for curative purposes or for studying the nature of the fluid. The medical instrument used for it is called aspirator. Pneumonia, sinusitis, pericarditis, meningitis, chronic abscesses, etc. are some of the conditions which call for aspiration. In pneumonia and similar problems, lungs and airway may become fluid filled. This blocks the flow of gases, causing difficulty breathing, hypoxia, and hypercapnia. And the problem can be temporarily solved by the aspiration of fluid with the help of an aspirator. aspirator has a soft tube connected to a suction pump. The nasal aspiration, the tube is inserted into the airway through the nasal opening and the fluid is sucked out with the help of a suction pump. So nasal aspirators are especially useful in children who are not able to forcefully blow their nose to remove a blockage in the airway. Then artificial respiration. Artificial respiration is a life-saving step or a first aid employed to ensure some degree of gas exchange when normal respiration fails and heartbeat continues. This has to be done within 8 minutes after the cessation of respiration. Some of the critical situations which demand immediate artificial respiration are choking, drowning, deep anesthesia during surgical operations, encephalitis affecting respiration, poisoning with opium, barbiturates, etc., carbon monoxide poisoning, electric shock, and rate at which artificial respiration is administered must coincide with the rate of normal ventilation, 16 to 20 per minute. Never forget that. 16 to 20 per minute. There are three major ways of artificial respiration, namely rhythmic pumping of air into the lungs through the respiratory passage, artificial rhythmic compression and expansion of the chest similar to natural respiratory movements, rhythmic electrical stimulation of the respiratory muscles. So, pumping of air into the lungs. This is the most widely used method. It involves the rhythmic supply of air into the lungs by means of a special pump operated manually or mechanically by a motor. Special apparatus has been devised not only to introduce air into the lungs but also to aspirate air from lungs. The simplest method of pumping air into the lungs is the mouth-to-mouth resuscitation. It can be employed when no special apparatus is available. In this method, a person administering the first aid periodically blows his expired air into the patient's mouth. This inflates the latter's lungs. The low oxygen content of the expired air is sufficient to ensure normal gas exchange. At the same time, the high CO2 content of the expired air stimulates the respiratory center of the same patient, which will definitely lead to the breathing. Then artificial respiration by Hooger-Nielsen method, rhythmic compression and expansion of the chest. This method is applied in several modified ways. The simplest of this is the application of pressure on the chest with hands with the rhythm of natural breathing. Air enters the lungs when the pressure ceases and the chest expands. Similarly, air passes out when pressure is applied and the chest and lungs get compressed. So, prolonged artificial respiration, often administered by means of special apparatus known as iron lung, which stimulate a metal cylinder into which the patient is placed. The front part of the cylinder has opening and patient's neck and head. This opening is sealed by a special collar. A special compressor periodically raises and reduces the pressure inside the cylinder. When the pressure decreases, chest expands and atmospheric air passes to the lungs along the respiratory tract. When the pressure inside the cylinder increases, chest and lungs get compressed and the air is expelled from the lungs. And the methods by which artificial chest movements are produced fall under two categories, manual and mechanical. Manual methods involve no mechanical devices, whereas mechanical methods are assisted by mechanical devices. The commonest manual methods include Hooger-Nielsen method, arm lift back pressure method, ALBP method, then Schaffer's method, Sylvester's method, and Eve rocking method. The commonest mechanical method include brag ball method and the resuscitator method, tang method. Next is about artificial ventilation. Ventilation is the process of air into and out of the lungs along the respiratory tract. In some critically serious conditions, lungs fail to function properly or stop functioning. As a result, ventilation becomes inadequate or impossible. In such situations, artificial ventilation is administered to save the life of the patient. Thus, artificial ventilation is a life-saving process which ensures free ventilation of the lungs and re-establishes the normal breathing rate and normal gas exchange. The automatic mechanical instrument employed in the artificial ventilation is called ventilator. Ventilators are of two categories: conventional or classical ventilators and high frequency ventilators. The breathing pattern and the breathing rate produced by the conventional ventilators are almost the same as those of normal natural breathing. On the other hand, high frequency ventilators produce high frequency and low intensity breaths. So the breathing rate will be much higher than that of normal breathing, natural breathing, but the depth and force of every breath will be much lower. So conventional ventilators are commonly used in home care and transport of the patients, and also in intensive care units and operation theaters. High frequency ventilators are usually used in patients whose lungs are very fragile or less elastic or air leakage happening. And heart lung machine, also called cardiopulmonary bypass machine or pump oxygenator is the machine which carries out the functions of both heart and lungs during open heart surgery. It pumps out blood and oxygenates throughout the blood. During open heart surgery, heart has to be opened, and therefore it cannot be used to pump out blood through pulmonary and systemic circulation. Hence, an external pump or heart machine becomes inevitable. If the lungs are not bypassed in the surgery, the anesthetist has to inflate the lungs nearly 10 to 15 times per minute for satisfactory oxygenation of the blood. This interferes with the operation. Hence, this practice has been abandoned now, and operations are done by passing the lungs with the help of external device for oxygenating the blood. It is called oxygenator or artificial lung. The external pump and oxygenator together form the heart lung machine. J.H. Gibson in 1953 of the Jefferson Medical College, Philadelphia, was the first to use the heart lung machine. Gibson's machine was very large, highly complicated, error prone, and difficult to handle, but still he just managed to use that and had several demerits also. In course of time, the heart lung machine technology was periodically advanced, perfected, and updated. The basic procedure followed in heart lung machine is as follows: deoxygenated blue blood is drained from the venous circulation by gravity siphon and then collected in a reservoir. From the reservoir, it is pumped through an oxygenator. Blood comes in close contact with the surface of the oxygenator. Now oxygen is delivered to space between the oxygenator and the blood. The hemoglobin of RBCs take up oxygen. Soon the oxygenated blood turns red. It is pumped to the artificial circulation through a tube with the help of an artificial heart. The major components of heart-lung machine include a set of pumps, an oxygenator, a heat exchanger, and several accessory components for control and regulation. The pump does the job of the heart by circulating the blood, and the oxygenator does the job of the lungs by giving preventions for gas exchange. The heat exchanger is used to gradually lower the body temperature. So lower body temperature, hypothermia, would reduce the metabolic rate, which in turn would reduce the oxygen recruitment during the surgery. The heat exchanger would be used again towards the end of the surgery to raise the body temperature to 37 degrees Celsius. Heart lung machine is geared and way such that it provides blood circulation at the rate of nearly 5 liters of blood in 1 minute with a pressure of 120 mmHg and blood O2 saturation above 95%. So external pump of heart lung machine is of lower type with no moving part in contact with the bloodstream. So it is called peristaltic pump. The movement of the rollers forces the blood forward in a peristaltic fashion. Pulsatile fashion, stimulating the pulsatory pumping action of the heart. So oxygenator is regulated in such a way that it can oxygenate up to 5 liters of venous blood per minute and raise the blood O2 saturation from 65% to about 95% over several hours without causing any serious damage to the blood constituents. Heat exchanger is usually a shell and tube type device. It has thin annular space with a hold up volume of less than 200 ml. Blood volume flows along the annular chamber and warm water flows through the inner tube. Oxygenated blood, venous blood, oxygenator, peristaltic pump, and warm water, and along with heat exchanger and an inner tube will be there.